All right, the next pattern of inheritance is sex-linked traits. So these notes will go ahead and show you how to do a cross when we're looking at a gene that is present on the X chromosome. So when a gene is being studied on the X chromosome, scientists use an X and an exponent, just like math class, to show the allele. All right. The classic example that we're going to go ahead and look at is fruit fly eye color. If you go ahead and take genetics as an elective your junior or senior year with Miss Ricciardi, you'll get to breed fruit flies. All right, so eye color is on the X chromosome. Chromosome. And the Y doesn't contain the gene. All right, so if we look here, we have 4 fruit flies. So we have a red-eyed female. So this is the symbol for a biological female. So the genotype would be X capital R, or X capital R, X lowercase r. The alleles that could be given when meiosis occurs would be X capital R or X lowercase r into the gametes that would be becoming eggs. All right. The red-eyed X capital R, X lowercase r is referred to a carrier because it carries the recessive allele. Now, this is the symbol for a biological male. So this is X capital R Y. And the only allele, the alleles that the male fruit fly can go ahead and give as the gametes, the sperm would be X capital R or Y. Remember, Y does not have any allele. We could have a white-eyed female. This would be X lowercase r, X lowercase r, and the only allele that could be contributed here would be X lowercase r. Or we could have a white-eyed male, X lowercase r, Y, and then again, the alleles that would be in the sperm would be X lowercase r or Y. All right. So in crosses with X-linked genes, the offspring are different depending on the alleles the parents have. So in this example here, if you cross a true breeding white-eyed female, so that means that it's homozygous, with a true breeding red-eyed male, we can't say that it's homozygous or heterozygous because it only has one X chromosome. This is the cross that you would end up with. You would have After meiosis, you would go ahead and make these 2 possible eggs. After meiosis, you would go ahead and make these 2 possible sperm. And then the cross that would end up would be a red-eyed female, and I denoted this by coloring it in red for you. Another red-eyed female. A white-eyed male. And a second white-eyed male. All right. If we crossed a true breeding red-eyed female with a true breeding white-eyed male, the possible eggs that we would have would be X capital R. The possible sperm that we would have would be X lowercase r and a Y. We go ahead and we do this cross. We have a red-eyed female. Another red-eyed female. A red-eyed male, and a red-eyed male. And so if you haven't gotten out colored pencils yet, this would be a good time to get out colored pencils so you could go ahead and you could see which phenotypes are linked with the corresponding genotypes. All right, so let's give this another try. So remember, when you do a genetics problem with an X-linked gene, you need to specify biological sex. So let's go ahead and talk about some mice. So mice with the recessive greasy allele have shiny fur. So I went ahead and I denoted this by using gray. So this is my recessive mouse here, okay? And mice with normal fur or not greasy is caused by dominant allele, capital N. So I showed that as using this tan color. Okay. The coat shininess gene is carried on the X chromosome. If a normal male is crossed with a carrier female, what % of the male progeny will have greasy fur? So we know that the normal, which trait allele is dominant? This would be normal. And this would be denoted with an X and a capital N. And then which trait would be recessive? This would be our greasy. This would be X and a lowercase n. So parent 1, all right, so a normal male is crossed with a carrier female. So let's do the male first. So parent 1, the phenotype is going to be normal. The genotype, we know that it's X-linked. So we know that this male mouse is going to have X capital N Y. And we know that the alleles that could be made through the process of meiosis would be X capital N or Y. The female mouse here, all right, is a carrier. So she is also normal. The genotype is X capital N, X lowercase n. So in this case, carrier would mean heterozygous for the female. She could contribute an X capital N or an X lowercase n. All right. So we have 2 normal mice. I'm going to go ahead and shade them in for us. I did some pre-shading. Okay. So let's go ahead and we'll put the male on the side here, X, capital N, Y, and the female is X, capital N, X, lowercase n. we go ahead and perform our crosses. We're going to have a normal female, a normal male, another normal female, which will carry the greasy gene, and a greasy male. So now our genotypic ratio is going to be slightly different because we need to denote which biological sex each is going to be. So we're going to have one capital X, capital N, capital X, capital N, one capital X, capital N, X, lowercase n, one X, capital N, Y, and one X, lowercase n, Y. All right. So if we're going to go ahead and talk about what they look like, we're going to have a normal female. We're going to have a carrier female. We're going to have a normal male. And we're going to have a greasy male. So 25% of all progeny are greasy, and 50% of all progeny are male, and we have 2 male here, so 50% of male progeny are greasy. Whoop. And the word progeny, if you haven't heard that before, equals offspring. They're synonymous with each other. All right, so the Y chromosome in most organisms have very few genes on it. All right. The most important gene on the Y is the SRY gene, and it is responsible for testes development. And if you go ahead and you take forensic science with me, your junior and senior year, we'll talk about the SRY gene when we go ahead and we do genetic genealogy to create family trees based upon cold cases. All right, so that is the end of our sex-linked traits video.